Some people once called much of our DNA junk. Well, this was a simple idea. It suggested that most of our genetic code was useless. It was like carrying around a backpack full of old, forgotten trash. This idea made life seem inefficient. If so much of our biological blueprint was waste, what did that say about us? It felt a little sad. It painted a picture of ourselves as cluttered attics, full of things that once had a purpose, but now just gathered dust. This simple label, junk DNA, shaped how we saw ourselves for a very long time. But what if the trash was not trash at all? What if it was a box of tools you forgot you had? New science is revealing a different story. It is a story of hidden purpose and secret missions. The parts of our DNA that looked quiet are not always sleeping. They are often hard at work, just in ways we did not understand at first. They are like the hidden support crew in a massive theater production. You see the actors on stage, but you do not see the lighting technicians, the stagehands, or the sound engineers. Without them, the show could not go on. How did we ever come to call our own genetic code junk? The story begins with a simple observation. Long ago, scientists discovered genes. They learned that genes are specific stretches of DNA. These genes hold the recipes for making proteins. Proteins are the tiny machines and structures that do almost everything in our bodies. They digest our food, carry oxygen in our blood, fight off infections. Finding these genes was a monumental breakthrough. It was like finding the most important books in a vast, ancient library. Scientists focused on these protein-coding books, but as they looked closer, they found something puzzling. The protein-coding genes made up a surprisingly small part of our total DNA, less than 2% of the entire genome. The other 98% did not seem to do anything. It did not contain recipes for proteins. It just sat there, stretching for millions of letters. Scientists did not know its function. Faced with this enormous mystery, some researchers gave it a simple, dismissive name: junk DNA. The term stuck. It was catchy. It seemed to make sense at the time. Science often works by focusing on what it can measure. The function of genes was clear. The function of the rest was a complete blank. You can think of it like early explorers mapping a new continent. They drew the coastlines, major rivers with great accuracy, but the vast interior they marked as unexplored territory. For a long time, junk DNA was the unexplored territory of our genome. This view persisted for decades. It was taught in textbooks. It became common knowledge. Many imagined ghosts of old genes and ancient viral infections. But as our tools improved, we saw the unexplored territory was not empty at all. The view of junk DNA began to crumble with new discoveries. As technology advanced, scientists could see what DNA was doing inside living cells. They could track which parts were being read and used, even if they were not making proteins. What they found was a shock. Huge portions of the non-coding DNA were incredibly busy. They were being transcribed into molecules called RNA. These RNA molecules were not just messengers. They were something else entirely, acting like conductors of a grand genetic orchestra. Imagine your DNA as a massive sheet of music. The genes are the notes the musicians will play. But music is more than notes. It needs instructions about timing, volume, and rhythm. It needs to know when to play softly and when to build to a crescendo. The non-coding DNA provides these instructions. Some small RNA molecules act like switches. They can find a gene and turn it on, or turn it off with precision. A single gene might be used in a brain cell but must remain silent in a liver cell. Other parts act like volume dials. They do not just switch genes on or off, they fine-tune their activity. They are called enhancers. An enhancer can be far from its gene, yet DNA folding loops them together. When it touches the gene, it tells the machinery to work harder, producing more of that protein. This is how cells ramp up production under stress. These discoveries transformed our understanding. The genome is a complex network. Non-coding regions are software running gene hardware. Tiny changes here can have huge consequences, from healthy heart to malformed one. Beyond acting as switches and dials, non-coding DNA has other surprising jobs. Some of it acts as a physical scaffold, an architect for the genome. Your DNA is incredibly long, about 2 meters if stretched from a single cell, yet it must fold into a microscopic nucleus. This packing is not random. It is highly organized. Certain non-coding parts act as anchor points, helping fold strands into functional shapes. Three-dimensional architecture brings far regions into contact. This is how an enhancer can reach the gene it regulates. The scaffold builds a functional city out of a one-dimensional road, creating neighborhoods where cooperating genes are brought closer. It also creates quiet zones, packing away unneeded genes. The shape of DNA is as important as its letters. A significant portion of non-coding DNA is mobile elements, sequences that copy and paste themselves. Long called selfish DNA or jumping genes, they were seen as parasites, and their jumps can sometimes disrupt genes and cause disease. They are a source of mutation and change, but also innovation. When they jump, they can carry regulatory DNA, dropping a new on switch next to a previously silent gene. This can create new traits for evolution to test. Our genome is filled with ancient elements tamed and repurposed, now permanent parts of regulation. Their secret mission was to supply new ideas, shuffling the genetic deck to keep life adaptable. There's another layer to this story, one that is even more dynamic. It is called epigenetics. This field explores how experiences and environment can leave marks on DNA. These marks do not change the sequence, A, T, C, Gs. Instead, they are like sticky notes or highlights placed on top of the code. They act as another set of instructions, what to read and what to ignore. Epigenetic marks are ghostly scribes in our margins. A common mark is a methyl group. On a gene or its control, it often signals "do not read," silencing the gene. Other marks unspool DNA to make it accessible. This system gives cells memory. Marks can be passed to daughter cells during division. A skin cell remembers it is a skin cell and not a muscle cell, even though both share the same DNA. Food, air, and stress can alter epigenetic patterns. Periods of famine or extreme stress can change methylation on DNA. These changes can adjust how metabolism and stress response genes are used for the rest of a person's life. Our DNA is not fixed destiny. It is a responsive script. Some epigenetic changes can even pass down a generation or two. A parent's experiences may subtly influence children's gene regulation. This is not about new genes, but inherited patterns of switches. The secret mission is adaptability, fast, temporary adjustments without waiting for slow evolution. So, what does all of this mean for you? Today, 2025, December 4th, it means the DNA within each of your cells is far more interesting than a simple recipe book. A living, breathing archive, a history book of our species' survival, a toolbox of instruments, a dynamic handbook, actively listening and responding to your life. The secret mission is not one grand purpose. It is the sum of many hidden, flexible, responsive jobs. Think of DNA as a vast library of what-if plans, contingency plans for your body, fighting off a new virus, repairing tissue after injury, adjusting metabolism if food becomes scarce. Most plans lie dormant. Non-coding DNA and epigenetic marks keep them silent until the right signal comes. A chemical message or environmental stressor. Then the right plan is activated. The secret mission is readiness. This changes how we think about health and disease. Many conditions are not caused by a broken gene. They arise from problems in the regulatory code, a switch stuck on, a dial turned too high. Instead of fixing a gene, we might adjust a switch, a nuanced approach honoring complexity. Let this inspire wonder. Your DNA is not just a legacy from the past. It is a partner in your present, holding potential for change and adaptation. a record of where you came from, and a set of tools for where you might go. In the end, the story of junk DNA is a powerful lesson in scientific humility. What we once dismissed as waste, we now see as a universe of hidden function. We have journeyed from cluttered attic to bustling, intelligent city. Non-coding DNA provides the power grid, communication networks, architects, memory keepers. Genes are the factories and workshops, but they cannot function without the vast supporting infrastructure. This shift is a triumph of curiosity over certainty. The secret mission is not mystical or alien. Evolution works in ways that are beautiful, messy, brilliant. It keeps options open. It saves old tools that might be useful again. It allows flexibility and rapid adaptation through layers of regulation. DNA is not a perfectly polished manuscript. It is a well-used journal. Notes, crossed-out ideas, brilliant insights, memories of past struggles. All of it tells a story and could be part of our future. We still have so much to learn. Scientists chart this territory daily, discovering new RNA molecules, discovering new regulatory functions. Some DNA may be true leftovers, but we can no longer assume the unknown is useless. The story teaches us to look closer, ask better questions, be patient with mystery. The universe inside our cells is as vast as the one outside. Do not picture a static string of letters. Picture a dynamic, living system. The conductors, the architects, the scribes, the movers, all working together. Your genetic code is not a sentence handed down. It is an ongoing conversation between your genes, your environment, your history. A call to wonder. What secret missions are hidden within us, waiting to be revealed? The answer is not junk. It is a fundamental part of what makes us alive.